most exciting and interesting information that I discovered as I started to investigate these sugars has to do with the ABO blood typing system that we use across the world. We have type A blood, type B, type O, and type AB. And surrounding each of these red blood cells, I have a legend down here at the bottom that you can see. 4 of these sugars, there are 8, as Angie mentioned, 4 that surround red blood cells. And they're arranged in a particular sequence, and there's actually 800,000 strands around every single red blood cell. That's almost 1 million strands of sugar around every single red blood cell. That's like a beard. The whiskers of the beard are these strands of sugar, and it covers the face of the cell, a forest or a shroud of sugars. Angie is type A blood. And these strands of sugars that are around it, around her red blood cells, have lots of different names. They can be called glycoproteins, glycolipids, cell adhesion molecules, cell surface sugar structures, cell receptors. Antigens is how the medical community refers to them. But I've taken one of those strands and I've blown it off to the side of the red blood cell. So if you look at type A, you'll see the 5 sugars that make up Angie's red blood cell. It starts with a molecule of galactose as the base right next to the cell membrane. And then the next 4 are in always the same order. And the last sugar that she has is a sugar called N-acetylgalactosamine. And that's the sugar on the very outside of the red blood cell. I'm type B blood. If you look at my blood sugars, it also starts with a molecule of galactose at the base of the membrane. And in fact, the next 4 sugars, those 4 sugars, are the same as type A. But my last sugar, the 5th sugar on the outside of the red blood cell, is different than type A. It's a molecule of galactose. And when I was studying this, it became apparent that there is no difference between type A and type B blood. They are the same. The red blood cells are the same. The sugars are pooled from the same 4. The sequence is virtually the same. The only difference between type A and type B blood is that last terminal sugar. But you know that if Angie's type A blood was transfused into my type B body, would my body have a problem with that? It will create antibodies, and they will go in and seek out and destroy every single one of those type A red blood cells. And the ramifications are as I will suffocate and die. And so when I learned that, it framed a question for me. And the question it framed was this: does the body know anything about these sugars if one sugar out of place has the power over life or death? That is a lot of power for a sugar to have in the body. Now, if you're type AB blood, 4% of the population is, you'll notice that you have the A sugar pattern and the B sugar pattern. I've shown that above and below the red blood cell. You're also known as the universal receiver, which means that you can receive blood from anyone because you recognize the 2 dominant patterns. You won't create antibodies because you're good with those other blood types coming into your body. Type O is the most predominant blood type. When you look at the sugars there, you'll see that there are only 4. Now, these 4 are organized in sequence in the same manner that type A or type B is. But because they only have 4 and they're missing the 5th offending sugar, that's why type O is known as the universal donor. They're the only blood type that can donate their blood not only to themselves, but to all the other blood types. So these are the folks solicited in blood drives. Now, all of these sugars coat the red blood cells, but they also cover all 60 trillion cells that make you. Every cell in your body is covered by these sugars. Here's what they look. This is a picture of a red blood cell. You see the membrane of the cell, the nucleus, and then on the outside you see the kind of filmy protrusions off the membrane. We used to teach that the membrane was smooth, but you see that it has all of these sugars coming off of it. This represents the operating system of the cell. Nothing gets in or out of the cell unless it connects and talks through these sugars. So this is vital for cell communication and for identifying the cells to the immune system. The National Academy of Science is a governmental body that's made up of folks who are Nobel Prize winners or you've been nominated by a Nobel Prize winner. They came out with a 200-page publication called Transforming Glycoscience. And in that publication in October 2012, they suggested that they want to take this science and they want to take it from the corner where it's been studied in a niche science called glycobiology. They want to make it mainstream. They want to push it down through the colleges and the junior colleges and into the high schools. They need career paths. They need the kids to be excited. They need folks to be into this field so that we can develop the instrumentation, a knowledge base of how these sugars work, because it turns out that these sugars represent the frontier of medicine. Now, I've taken 4 quotes from that 200-page publication. The first one at the top states that every living cell on planet Earth is covered with a dense and complex array of glycans, or these sugars. So there's a shroud of sugars covering every cell, not only all human, but all animal and all plant. Every living organic cell has these sugars, and they're the operating system for the cell. And the second bullet, they stated every disease that affects humans significantly involves these glycans, or these sugars. They play a part in all of the diseases that are being manifest. And so this is why it is the frontier of medicine. We need to understand what the biomarkers are for these disease states. In addition to every major disease, they're involved in almost every biological process that goes on in your body. So all of those chemical reactions that are going on, most of them involve these sugars. The last bullet down here, I have a picture that goes along with it, and you see these tree structures that represent these sugar organized on these cells with a cell antenna system that looks like trees. They're bush-shaped. They're complicated structures. If I were to eliminate any of those, just part of those class of sugars, the cell will die because it can no longer identify itself. It can't tell the body its nutrient needs. And so these sugars are the operating system. I can actually go in and take the nucleus out of the cell, denucleate it, and the cell will continue to live for months. It can't reproduce because I've taken its DNA away, but the cell will live. If we disrupt any of these sugar structures on the cell, on the outer surface of that cell, the cell will die immediately because it cannot communicate. What's exciting about this is that a national organization has put together this information about glycoscience and has identified it to the world, that this represents the frontier of medicine. And with the emphasis on this education of learning more about how these sugars are involved in our bodies, we're excited to see where the science will lead. And truly, it does represent the frontier of medicine. In this segment, we'll talk a little bit about how the cells are structured and how the sugars interact with these cells. On this picture on the left, you'll see the nucleus of a cell, the cytoplasm, and then the cell membrane. Again, in biology textbooks, this membrane used to be a smooth surface, but you can see that it's very hairy with a lot of these sugars protruding from the cell membrane. On the right, you'll see an illustration, a picture of these cells. There are 2 of them, one cell membrane kind of arcing down to the left, and the other one on the top. In between that, you'll see the glycocalyx. You'll see that those sugars are connected to each other. The cells are connected via those sugars. When they are doing this, we call them cell adhesion molecules. But also, in addition to holding you together, and this is how you're held together by these sugars, signals and communication takes place across these cell antennas. So the cell on the top will communicate something to the cell on the bottom, but it must do that through those sugars. They serve as the antenna system for the cell. This illustration on the left shows some spherical cells. The hairy protrusions are these glycoproteins. You'll see the 2 lower cells are holding on to each other, again, as cell adhesion molecules. The cell at the top is kind of an abnormal looking cell. It's devoid of those glycoproteins. It's not able to communicate with the immune system, and so things are going wrong with it. It's barely hanging on to the cell below it. When it loses those glycoproteins, it will no longer be anchored to those cells on the bottom. It will be free to transverse through the body, through the circulatory system, through the lymphatic system, and if it's a cancer cell, it will lodge somewhere and become a secondary cancer. This is called metastasis, when cells lose that adhesion and are no longer anchored to the tissue. On the right is an illustration of a cell. You can see the green part is the protein going through the cell membrane, and then the purple pinkish color are the sugars. So again, they branch out in bush-shaped structures. Good guys and bad guys connect here to these sugars. White blood cells will connect and identify whether this cell belongs in the body or needs to be destroyed. Bad guys like bacteria will dock here and release a toxin. Viruses are more tricky. They'll actually kidnap the manufacturing plant within the cell to make copies of themselves. Hormones also attach here at this point. So this represents the operating system of the cell. Nothing gets in or out of the cell except via those sugar structures. There are a lot of models out there that talk about sugar, and we've learned a lot in recent years. But one of the theories that we had that turned out not to be true is that we thought sugar was just used for energy production. And you took sugar in to increase your metabolism and to get energy. While that's true, we didn't know about these other sugars. These other 8 sugars are not fuel sugars. They're not metabolized. They're important for cell-to-cell communication, for the immune system to do its job. On the lower right, you'll see a macrophage, a white cell that is actually moving towards bacteria. And those sugars allow it to sense who belongs in the body and who does not. In this slide, you'll see another model that said that all sugar was changed to glucose. All you needed to eat was glucose, and the body would take care of everything else. Well, the French discovered that that was not true. Your body is actually looking for these other sugars. In the gut, there are cell receptors looking for that. In fact, they were so impressed by what they found in their study in 1998 that they said, this represents a new class of nutrient. Similar to vitamin and mineral, we now have something called glyco or sugar nutrients. Again, they weren't broken down from glucose. They're not the same as table sugar. They don't impact blood sugar negatively because they're not metabolized. They build structures. The immune system must have them. The macrophage that you see in that picture, again, needs to have in order to sense who belongs in the body and who does not. This particular slide shows the communication system power of these sugars. We originally thought it was protein-based, but proteins don't have enough combinations to make enough letters in the sugar-coated vocabulary that the immune system needs. Because when you cut your finger, the immune system somehow knows that the cut is here, not on your index finger or your pinky finger or on this hand or on your foot. It directs the forces to the right location, initiates the inflammatory response, and takes care of you and protects you. How does it do it? Well, it does it with these 8 sugars. And these 8 sugars are actually hexagonal in nature, and they are 3-dimensional. So they're much more complex, and scientists have estimated that there's somewhere between 1 × 10 to the 14th and 1 × 10 to the 15th chemically unique letters in this sugar-coated alphabet. Now, 1 × 10 to the 14th, I've written it out for you, a 1 followed by 14 zeros. It's pronounced 100 trillion. And 100 trillion is huge. If I take off the first 6 digits and replace them with a 1 followed by just 9 zeros is a lot smaller. And that number is pronounced 1 billion. And if I were to ask you how long a billion is, I would use seconds. Because seconds go by pretty quickly. And if you had a billion seconds, how long is that? And when you calculate it, it turns out to be 31.7 years. 31.7 years is what a billion seconds is. And we're talking about a communication system that is 100,000 billions. That's a lot of letters, which make a lot of words, which make a lot of phrases, which communicate chemically the activities that we know our immune system does to protect us. And when the immune system doesn't communicate properly, science has shown that sugars are missing from those cell surface sugar structures. And if I were to represent a normal immune system with a horizontal arm, what happens when the immune system is like this is it protects you, it takes care of intruders that come in the front door, it takes care of problems going on in your house inside. But what happens when it stops doing that? When it starts to fall down on the job, we call that degenerative. It knows what to do, but for some reason it gets lazy or miscommunicates and no longer takes care of business. If the immune system goes this way, it goes up, it becomes hyper, it starts to do extra things that it's not supposed to do. Instead of defending you, it decides to take on another task and attack you. It's not supposed to attack you. 4 months ago it didn't attack you. Why does it change its mind and start to attack you now? So when you have a degenerative to autoimmune, does that cover a lot of territory? According to the National Academy of Science, every major disease affecting humans involves these sugars. And when you have a degenerative or autoimmune issue, sugars are missing from those cell surface sugar structures. Now this video that's going to play shows a neutrophil that's actually chasing bacteria through a group of red blood cells. You can see a little aura around the red blood cells, and that's those sugars extending. And you can see that the white blood cell pushes its way through but never gets right in onto the cell. It never attacks any of the red blood cells, but it chases after that bacteria, and no matter where it goes after it until it gets it and takes care of it. And so what's cool is that is what you don't see. Because of those sugars, the macrophage is able to identify what belongs in the body and what does not. And if you don't belong in the body, it will take you out. In summary, what I hope that you've learned from this is that there are these cell surface sugar structures on the outside of all cells. They're needed for the proper recognition and the function of those cells. Immune system cells come in and they read those sugar cells, sugar structures on these cells, and they determine whether you belong in the body, if you need support, or if you need to be taken out. And they will take action based on what they read. When cells turn against the body, as in degenerative and autoimmune, science has shown that sugars are missing from those cell surface sugar structures. This is a macrophage down at the bottom right attacking a cell. From Rockefeller University in 2007, they removed a sugar from a protective antibody, one of your defensive immune system components. And when they took a sugar off of this antibody, instead of the immune system defending you, it turned around and attacked. So we understand the mechanics of autoimmune. Now there are many studies that have been done with these sugars over the years. Typically they've been done in animal trials, and there are amazing things that you can see. But people will sometimes ask me, if they're so amazing, how come they never transition them over to the gold standard, double-blind, randomized, controlled crossover kinds of studies that healthcare professionals would watch and read? Well, simply for one reason is what I've come to understand, and that one reason involves time and money. It takes a decade to bring a drug to market in this country, and it costs $5 to $6 billion. If you can't recoup your investment because you can't make these sugars synthetically because they are too complicated, then why would you ever pay to do that? And so folks are trying to make these things synthetically. And the fact that they exist, these sugars exist in nature, it's not the job of the pharmaceuticals to tell you. They don't do the natural kinds of things, they work with synthetics. So, but since 2011, there are some of these human trial studies, double-blind, randomized, controlled, crossover kinds of studies. So we can now document improvements, statistically significant improvements in many cases, for immune system function, for cognitive, for memory, concentration, attentiveness, mood, irritability, digestive, and cellular communication. So they're coming, and there's a great hope as they come. This research that was published in the European Journal of Clinical Nutrition debunks an old theory that was out there, again, another belief that turned out not to be true, Scientists thought that it wasn't possible to influence the body's production of these cell surface sugar structures by changing what you ate. That you were a genetic victim, that your DNA just did the mapping, and that's what you got. So if it wasn't correct, so sad, too bad. You couldn't do anything about it. But with this study where they gave these 8 glyconutrients to humans, they showed that you could induce statistically significant changes in those cell surface sugar structures with no adverse effects, because there was no toxicity. It was just food. And that reminded us that the body, you can have genes, you can have these tendencies, but they don't have to be expressed. And so the availability of nutrition, the surrounding and the environment of the cell, all influence what genes get turned on and turned off. There are little manufacturing plants within each cell, and they manufacture these structures. And when they go down the conveyor belt, you want to have those ingredients in place so that the body can make these structures. Because if that ingredient is not there, then you can't put it in place when the body calls for it. And the body can't stop. It can't wait and order out. It has to keep moving, because to stop would be death. So it keeps moving, it substitutes, it skips it, and this cell antenna that gets put out may be correct or it may be defective. And so structure is function. If you don't build that structure properly, then the body doesn't function properly. You make 150 to 500 billion new cells every day. Every second of your life, you make 1 million cells. You want to have the ingredients so that the body can make these structures so that you can have the proper function to have optimal health.